Oxidative phosphorylation by Jaime Coberti. Oxidative phosphorylation is the climax of energy-yielding metabolism in aerobic organisms. All oxidative steps in the degradation of carbs, fats, and amino acids end at this final stage of cellular respiration, in which the energy of oxidation drives the synthesis of ATP. Oxidative phosphorylation accounts for most of the ATP synthesized by non-photosynthetic organisms under many cases. In eukaryotes, oxidative phosphorylation occurs in mitochondria and involves protein complexes in the mitochondrial membranes. This is known as the chemiosmotic theory and has been accepted as one of the great unifying principles of the 20th century. It provides insight into the processes of oxidative phosphorylation and photophosphorylation in plants. The mechanism of oxidative phosphorylation has three components. One is that electrons flow from electron donors. Two, the free energy made available by this downhill electron flow is coupled to the uphill transport of proteins. And three, the transmembrane flow of protons back down their concentration gradient. With electrons flow from electron donors, they do that through a chain of membrane-bound carriers to a final acceptor with a large reduction potential, oxygen. With the downhill electron flow being coupled to the uphill transport of protons, they do this with conserving the free energy of fuel oxidation as a transmembrane electrochemical potential. And for number three, the transmembrane flow of protons back down the concentration gradient, they do this through specific protein channels that provides the free energy of the ATP synthesis, catalyzed by a membrane protein complex. Oxidative phosphorylation begins with the entry of electrons into the series of electron carriers called the respiratory chain. Most of these electrons come from the action of dehydrogenases that collect electrons from catabolic pathways and funnel them into the universal electron acceptors known as NAD or NADP, or the flavin nucleotides, FMN or FAD. It is important to not get this process of oxidative phosphorylation confused with substrate-level phosphorylation. As in substrate-level phosphorylation, the formation of ATP and 3-phosphoglycerate, as for example, involves a direct phosphate transfer and is not considered to be an oxidative phosphorylation regardless of the energy that is released. Oxidizing the reduced forms of the coenzymes FADH2 and NADH is only possible in the presence of ADP, leading to generated ATP. The image shown depicts the oxidative reduction potentials for NADH and FADH2 reducing to oxygen. As seen, in both cases the combination of potentials is positive. This indicates that both are spontaneous, yielding energy. The following animation I've created will display the significance of the electron acceptors in the ETC. When we first look at complex 1, known as NADH oxidoreductase, as can be seen by electron NADH, NADH will oxidize and help with the transfer of 2 electrons to FMN. This will complete the downhill movement into complex 1 until 2 protons come together to ubiquinone to be released as ubiquinol and transferred to complex 3. Then, in complex 2, with the succinate dehydrogenase, FAD will accept 2 electrons from succinate to become FADH2 and transfer those electrons to iron sulfur centers to finally become ubiquinone to make ubiquinol, QH2. Okay, now quiz time. Yes, I know. Get your eye clickers ready. You have 30 seconds. The transmembrane flow of protons back down the concentration gradient through specific protein channels provide the free energy for synthesis of ATP, catalyzed by a membrane protein complex, ATP synthase, that couples proton flow to the phosphorylation of ADP. The chemiosmotic mechanism for ATP synthesis in mitochondria, shown in figure 19.1, states, electrons move spontaneously through a chain of membrane-bound carriers, the respiratory chain, driven by the high reduction potential of oxygen and the relatively low reduction potential of the various reduced substrates that undergo oxidation in the mitochondria. The processes of oxidative phosphorylation and the electron transport chain are closely coupled. Oxidizing the reduced forms of coenzymes, such as FADH2 and NADH, are only possible in certain positions. They are only possible in the presence of ADP, and the oxidations couple with ADP, and that is how you transform to ATP. Hence, phosphorylation.